Hi and Assalamualaikum. My name is FAIZAH binti ZAINAL ABIDIN and my matrix number is 208074. I'm going to do the hands-on assessment for BMY3201 and I'm going to demonstrate step by step the technique for streaking for single isolated colonies. The streak plate technique is the most popular method for isolating specific bacteria from a sample containing a mixture of microorganisms. The technique essentially dilutes the number of organisms and reduces their density. The sample is diluted by streaking it across the surface of the agar plate. When these lone bacterial cells divide and give rise to thousands and thousands of new bacterial cells, an isolated colony is formed. Pure cultures can be obtained by picking well-isolated colonies and restricting these on fresh agar plates. Pure colonies is an individual colony that is transferred to another plate to produce a pure culture consisting of one type of bacteria. In this experiment, we need agar plate, mixed culture, inoculating loop, and a marker. Using a marker, label the quadrant 1 through 4 on the bottom of the petri dish. Before we inoculate the culture, aseptic technique is carried out by flame the loop and the neck of the tube by rapidly passing them through flame at once. Turn the petri dish over and place a loopful of culture on the agar surface in quadrant 1. Using the edge of the loop and holding the loop at a shallow angle so as not to gouge the agar, quickly spread the bacteria throughout the quadrant. Reflame and cool the loop and turn the petri dish 90 degrees. Then touch the loop into an area that has been streaked in quadrant 1 and drag it across the agar into quadrant 2. Make sure the loop do not touch the previous area. Reflame and cool the loop and again turn the dish 90 degrees. Streak the bacteria into quadrant 3 in the same manner used for quadrant 2. Next, without reflaming the loop, turn the petri dish 90 degrees and drag the bacteria from the corner of the quadrant 3 to quadrant 4 using the wider streak. After you have done the experiment, a aseptic technique will carry out by flaming the loop once again in the flame. Lastly, incubate the plate in an inverted position for 48 to 72 hours at 25 degrees Celsius. This is the expected result for the mixed culture of S. aureus and E. coli using streak plate technique. From this experiment, we can observe and distinguish the two colonies. For the observation, we can see that both have the same form, elevation, and size, which is circular, raised, and small, but different in pigmentation. For isolate 1, it has a yellowish white, which can be concluded as S. aureus, while for isolate 2, it has a white color, which can be concluded as E. coli.